Hello, everybody. This is Team Domino Lils, with Cash, Tyler, and Robert. I'm going to be going over question one with you. So it's just a lot of reading, but try to describe what I can describe. So the first one talks about which term or is described or is an example of a regulatory protein. So examples in the are repressors and activators, and also CAP is part of it as well. For part B, which term describes or is an example of a small effector molecule? And those ones are inhibitors, allolactase, cyclic AMP, and an inducer. And so those ones are those. And then for part C, it says which term is an example of a sequence found in the DNA. So we're looking actually within the operon or within the mRNA or whatever we're looking at. So we have +1, -10, LAC-Z, LAC-A, and the operator sequence. And so which of the, and then for part D, which of the above is not involved in live operon, and we put they are all involved in it. So it's kind of tricky there. For part E, it says what example of this is an example of a structural gene. And for this one, we illustrated that we thought it was going to be LAC-A and LAC-Z.
All right, I'm going to be doing number 2. So these are the fill in the blanks. So for letter A, it says the LAC operon is regulated by 2 inducers, and those are allolactose and CAMP, cyclic AMP. And B, it's talking about certain binding sites. So it says the binding site for the sigma factor in RNA polymerase is called the promoter site, the LACP. And then the site for the binding for the activator that regulates the LAC operon is the CAP, the CAP site. C, the enzyme adenyl cyclase converts ATP into CAMP. And then letter D talks about the -35, -10, and +1 sequences, which are found within the LAC promoter again, LACP. E is, it says the blank gene encodes the LAC repressor, and that's the LAC-I gene. And then the blank gene encodes the lactose permease, and that is the LAC-Y gene. F, it's talking about this can regulate gene expression of bacteria by causing transcription to terminate prematurely. We found that was attenuation, which we found in instructive notes is specifically when transcription terminates prematurely, it's called attenuation. And G, an E. coli strand that contains 2 copies of the LAC operon is called the merozygote.
Hey everyone, so I'm going to do problems 3 and problem 4. I have my phone here just to kind of help me have some notes on how we got these answers so you can understand as well. So for 3A, we said that the cell will not turn blue, and the reason for that is simply there's just no IPTG, which starts the whole process of transcription. For B, we also said it will turn blue. I marked in red, hopefully you can see that, but it will turn blue. This is the only one that does turn blue out of all of them. The reason for that is the IPTG will bind to the LAC repressor and begin the whole process of transcription of the LAC operon. For C, again, it will not turn blue. The reason for that is there was no X-gal present, and we need that to turn the cell blue, because once metabolized, that's what gives the cell a blue color. For D, it will also not turn blue. The reason for that is since glucose is present, it's going to be preferred over lactose, which is going to bring the CAMP levels down really low, which is going to prevent the CAP from leaving the CAP binding site, which starts as one of the initiators, I guess, or starts the whole process of the transcription. E, again, will not turn blue. Kind of the same reason as before. There's glucose presence. Specifically, though, it doesn't really make a difference that X-gal was or was not present in either D or E, because glucose is going to be preferred to prevent the beginning of the transcription of LAC operon. Now for 4, for A, we got inducible, and the reason for that It's pretty simple. Everything is fine. There is no cause for concern. Everything was exactly the way that it was supposed to be. B was non-inducible. The main reason was the promoter was defective. C is where it starts to get interesting because we start to have the plasmids. So for C, we got constitutive, meaning that it'll never turn off. It'll constantly be running. And the main reason is the lac operant on the chromosome has a defective lac promoter region. And so it's constantly just going to be running. It's never going to be turned off by the repressor. For D, we said the cell will be inducible. And the reason for that is from having a correct lac repressor that can bind prime plasmid cannot produce a correct lac repressor. So again, that makes D inducible. For E, we got that the cell will be constitutive. And the reason for that is the cell has a correct repressor on the F plasmid. However, the F prime plasmid also has a defective operator. So the F prime plasmid will constantly be transcribing the lac operant.
All right, I'm going to be doing number 5, the last question. Very similar to number 4, in principle. So we're talking about specific E. coli genotypes and if they will, how much yellow they will produce based on their genotype. And so for letter A, we got that this E. coli gene would have no yellow produced if lactose was present and not present. And that is because the promoter region is defective, which means that the RNA polymerase cannot bind to the promoter, so it will never be transcribed. B, we got that this E. coli gene will always produce the yellow either in the presence of lactose or not in the presence of lactose. This is because the operating sequence is defective, which means that the repressor cannot bind to the operating sequence, so it is constitutive, so it will always be transcribed no matter what. C, now these ones we start getting where we have 2 chromosomes, a chromosome and a plasmid. And so C, we saw that this first chromosome is all wild type, so it's fine. This one has a defective operator sequence, so it's going to be transcribed all the time, even when there's no lactose present. So that's why we put a 1. And so when lactose is present, they'll both be transcribed. So it'll have 2, a lot of yellow. So that is what we put represents the number 2. D, which here we have defective repressor. This repressor here cannot bind to the operator sequence. And here we have defective promoter. So the plasmid will never be transcribed, even when there's lactose present, because RNA polymerase can't bind here. But it does have a good repressor, a wild type repressor. So this repressor can bind to both of the chromosomes, the chromosome and the plasmid. So we can bind over here. So we put 1 when there is lactose present and 0 when there is no lactose present. For letter E, this chromosome has a defective repressor in that it cannot bind to the operator site. But this one down here is the IS, and it's a defective repressor, but it can bind to the operator sites, but it cannot bind to allolactose. So here, since this repressor can bind to both of these operator sites on both chromosomes, the chromosome and the plasmid, but it cannot bind to allolactose. So even if there is lactose present, it won't be able to bind to the repressor and remove it from the operator sequence. So these are both non-inducible. So that means we put 0 for when there is lactose present and when there isn't lactose present, we got 0 for both. So those are our answers for the lesson 12 group problems, and thank you for watching.